Hi. Previously, we've discussed the topic of sedimentation, Sedberg's equation and value, taking the example of the sedimentation velocity and centrifugation velocity of 70S ribosome, and how in centrifugation, separation can outrun diffusion. For today, we're going to discuss blood centrifugation by the classical method and see how not just laboratory centrifugation of the kind we would practice maybe in a basic biology lab, but also in a clinical setting, this might be relevant. So what you're looking at here is a typical electronic centrifuge, and the separation of saturated blood into plasma, buffy layer, that is to say the white blood cell and platelets. This we can call buffy layer. And this is the RBC layer. This is because blood itself consists of three major centrifugable components, or those that separate on centrifugation. The plasma, which is sort of a clear fluid, the buffy layer, which contains the red blood cells and platelets, and the red blood cells, which form a very large proportion. To remind ourselves, red blood cells themselves are discoid structures which have a diameter of between 6 and 8 microns. And indeed, you will find that this size of the red blood cell is itself used as a metric for health. And in contrast, white blood cells can be large, reaching 12 to 15 microns in diameter. They are also nucleated as opposed to RBCs. At least mature RBCs in humans are enucleated, they lack a nucleus. The granules I'm drawing in the white blood cells are typically, depending on the type of granule, neutrophils, basophils, etc., they arise from the specific morphology of nucleophilic stain. Now, blood itself is not static in terms of the fact that it flows. And the flow is non-Newtonian. This is mostly because blood consists of these two primary components, cellular components and plasma, which is a mixture of macromolecules, ions, and water. The cellular components consist of RBCs primarily, the largest population, WBCs, platelets. The red blood cell size is also associated with a volume, which is usually in the range of 98 micrometer cube, and a surface area of 130 micrometer square. During this flow, you may observe that these discoid red blood cells form a sort of stack of dishes. This is also referred to as rouleau formation. It indicates stagnant RBCs, resulting in the stacking. The alternative is that they align in the direction of the flow. So if this is the flow direction, then the RBC is approximately aligned to the flow. What is the typical content of RBCs in human blood? Concentration. We find that on an average, the male population has between 4.5 into 10 to the 4.5 to 5.5 into 10 to the power 6, and female population on an average 3.8 to 4.8 into 10 to the power 6. The units is cells per microliter. Microliter is 10 to the power minus 6 liters. The mean corpuscular volume is a volume that is somewhat comparable to what we discussed here. It's a 98 micrometer cube, which translates into between 78 to 94 femtoliters per RBC. You will find it convenient to remember that the orders of magnitude go like this: 1 10 to the power 0 liter, 10 to the power minus 3 liter is 1 ml, minus 6 liters is 1 microliter, minus 9 liters is 1 nanoliter, minus 12 pico, hematocrit, which is often something reported in a typical complete blood count, is the volume of RBCs to the total blood volume. Since this is a dimensionless number, just a ratio, in theory, a centrifugation or even just sedimentation, which allows the blood to settle down, can be used to estimate hematocrit percentage by taking the ratio LR to L from such a standardized tube. There are many references on clinical methods and physical and laboratory examination, and Billet is one of the books that, if you're interested in reading more, you can go back and look. In fact, as it turns out, the hemoglobin concentration and the hematocrit, which can be reported also in percentage terms, multiplied by 100, for females and male humans is reported to range for hemoglobin of 12 to 16 and 14 to 18. This is in grams per deciliter, whereas hematocrit is 36 to 48 for females and 40 to 54 for males. Many of the modern RBC and hematological tests are done using a Coulter counter, that is to say automated, which has many advantages in terms of throughput, accuracy, objectivity in identification, and a few others. But for laboratory demonstrations, we still use conventional methods to try to understand the principle. And the straightforward way that in the past, when we've done experiments, has involved taking a volunteer, that is usually me, to subject themselves to autophlebotomy. A small pinprick is made, tiny amount of blood is released, that droplet from the puncture wound in the peripheral blood vessel is taken by capillarity into a special tube that is made for these sort of experiments. These are thin capillary tubes that are previously heparinized in order to prevent coagulation. And by applying it to the wound, the blood is sucked in. Then this material is centrifuged and the packed volume and plasma can then be observed with the buffer coat in the middle. This then being the L total. So H then becomes L pack by L total into 100 to give you percentage Hematocrit. We will come back to this in the context of low cost centrifugation in a paper that we will discuss by Bamla et al.